Let's talk about the principle of affinity chromatography. This is done by keeping the various biological functions as well as the biological interactions of the molecules to be tested in mind. The interactions may be antigen-antibody interactions, enzyme-substrate interactions, or receptor-ligand interactions. These interactions are characterized by their specificity, allowing for the selective retention of the target biomolecule while other components in the mixture are washed away. We have a sample solution consisting of the unwanted biomolecules as well as the protein of interest. In order to isolate the protein of interest, we must know the properties of the protein and know which molecule it binds to. This molecule, which might be an antibody, receptor, or an enzyme, is then immobilized on a stationary phase. The stationary phase consists of a solid matrix to which an affinity ligand is covalently attached. Common matrices include agarose and polyacrylamide. This is known as column preparation. Once the column has been prepared, the sample containing biomolecule of interest is applied to the column, allowing specific binding to occur. This is called as the loading of the sample. The unwanted biomolecules pass through the column without binding and are separated out of the sample, leaving behind only the molecule of interest. The elution buffer may contain chromatographic agents or affinity ligand. Next, an elution buffer is added. The elution buffer must effectively disrupt the specific interactions between the target biomolecule and the immobilized ligand by change in pH or by changes in ionic characters. These changes cause the target molecule to dissociate from the immobilized ligand. This releases it into the elution buffer. This is called as elution. Histag is a stretch of 6 to 9 histidine residues. Histag is the most commonly used tag used for the separation of proteins. The tag can bind to both the N-terminal and C-terminal of the protein. The stationary phase used here is most commonly a nickel nitrilotriacetic acid bead on a resin matrix. The protein that has been tagged using histag is present in the sample NAD is loaded into the column. The histidine residues form a coordination bond with the nickel ions, binding protein to ligand. Imidazole buffers are used to elute out the protein of interest as imidazole competes with the bound protein, disrupting its attachment to the beads. Affinity chromatography has high specificity for selective purification. It is highly efficient as significant purification can be done in a single step. It is applicable to a diverse group of biological molecules.